Thank you, Sylvia. Welcome, everybody, also from my side. Good afternoon. I'm happy that people join our webinars in Basel and also online. I want to give a warm welcome to today's speaker, Professor Mark Rubin. He's gonna introduce to us today the SOCIBP project, which is actually short for Swiss Oncology and Cancer Immunology Breakthrough Platform for Precision Oncology. We're very happy to have you, Mark, today. Mark joined the University of Bern as a professor and director of the Department for Biomedical Research in May 2017, actually coming from New York, from Cornell, where he was also the founding director of the Precision Medicine Institute at Weill Cornell. In 2019, he was also appointed president of the newly founded Bern Center for Precision Medicine at the University of Bern and the Inselspital. Thank you for coming again, Mark. We very much look forward to your talk.
So good afternoon. So I'm going to talk a bit about our program as part of the SPHN program. I just want to give a little context with regards to where this fits in precision medicine. So I'm going to be talking about oncology and genomics data. But in fact, if you take a step back, whether it's genomics data or whether it's other type of data, it's really important in precision medicine that we find ways of connecting patients, patient data in both a small way, so for the individual patient, as far as taking care of that patient, how we think about that, but also in a larger way, how we think about that individual, where they fit in a context. And so, you know, there's a number of challenges. And so what we're going to talk about today, and just for briefly, and then we'll have questions, will be a program that's job is really to take care of managing genomic data in the context of cancer care. So I like to think of precision medicine in these circles where we're connecting data. It's genomics and patient data and electronic health data. And how do we do that? We're really fortunate because whether just by luck or well-planned, the Swiss Personalized Health Network had two groups that came together to collaborate. And so our group was funded to do genomics. And then there was a SPO program, which looked at oncology side. So we have clinical oncology and genomics working together. And that's been a great partnership and continues, I think, to build our ability to think about really at the patient level, but then also Swiss-wide, how are we going to take care of patients in the field of oncology. These are the sites and the individuals involved. And at the outset, we wanted to have a project that would also ask a specific question. So I'll tell you a little bit about the specific question. And that's part of the EHT Domain funded PHRT program grant that's led by Gunnar Retsch and George Kukos at both at the ETH Zurich and at the Ludwig Institute in Lausanne. So what were our goals for this project? And you heard in the introduction that before coming to Switzerland, I had founded and developed a precision medicine institute in New York. And over approximately 5-year period, we learned a lot about how to develop pipelines to analyze genomic data for cancer care. And I think the importance of that was that we needed to do the genomics work at a level that would be not research, but would actually be at a clinical level that we would use for research, but then eventually also clinical. And the reason for that, which was more complicated, more expensive, it required us to go through a lot more regulatory hoops, was that we felt that if we have a system that works from the very beginning in a clinical setting, then it would allow us to obviously do research, but also then to make a very simple transition into the clinical side. And so perhaps a little bit naive, but the idea at the very outset was that we would develop a research grade tool that we could use. So I'll describe that a little bit. And the reason why I say naive is because I was just coming to Switzerland, so I don't know anything about Swiss regulations. I know about New York regulations. I know about the FDA in the United States. I don't know very much about European and Swiss, so I had to learn about that, and we're learning, I think, as we go. The analytic pipelines would need to be set up. And I think there's another important lesson that we've learned is that what we started with 5 or 6 years ago in New York, and my assumption was that that would be pretty good, really is almost, as I wouldn't say not relevant, but I think the relevance changes over time because the tools improve. And I think there's a real movement for more community-based tools. So that is open source tools that can be used by a broad community in the oncology field and other fields. And I think it's really important because when you imagine this as something you're going to set in stone and say this is the system, in reality it's going to be dynamic. And it's a little bit challenging in the clinical setting because in the clinic you want to freeze a pipeline for clinical care, but you can't change it every day. It's not a research project. And so that's obviously an important issue. And then obviously in order to do this, we'd have to test it in real patient samples and see how it functions and think about how that would work. And then develop ways that we could report this information. So one of the other, I think, important lessons I learned in New York was that when working with genomic data, which is by nature complex, the reporting of this information is also complex. And it's not just complex for the patients, it's also complex for the clinicians who take care of the patients. Even sophisticated oncologists or surgeons who are involved in the process may not understand the information that's coming off of these next generation sequencers. They may not understand the wording or the implications. And so it's actually very important that we think about ways to communicate this information. So standardizing reports. And then also, as we're doing this, and one of the, I think, the big investments in any precision medicine program is not what you're going to use the data for today per se, but also how is it going to be used 5 years and 10 years from now, because you're creating a huge database. And my colleagues across the street when I was in New York at Memorial Sloan Kettering now have a database for their oncology patients that I think is upward of 70,000 patients that have been analyzed in their single institution. And so you can imagine the value of that is that anytime an individual patient comes in, you can say, hey, have we seen this before? And if so, what happened to that patient, or what did it look like, or how does that patient compare to other patients? And there are some very nice examples, without going into specifics, where rare cases, so 1% of a population, or less than 1%, might have an alteration. So, but you really need large numbers to appreciate that. So that, I think, is very important. And then, in addition, how do we process this data at all the different hospitals? How do we accredit the labs for this? And then, if I have time, I'll just very briefly mention the PHRT part, which is to look at how do we use this data in the context of a specific trial, which was immuno-oncology. So these are the 3 work packages. I sort of just described them. I'll just go very briefly just to say that, step 1 is to look at the pipelines and figure out how we can optimize the pipelines and get data off of it. Step 2 would be to develop these reports. And obviously these reports would be more of a molecular pathology report, but obviously they could be integrated with the clinical information that the SPO group is gathering, as well as pathology data. So I can mention that briefly. So pathologists are also going to be gathering data. And then the last part is work package 3 here, which is very important, which is how do we share the data? And there are different ways that we want to share the data. So as a researcher, I'm a lab-based researcher. We do basic science in my lab, and the people in my lab don't want to become computational biologists, but they want to have access to the data. So there are a number of ways that we can share the data with researchers. And then for the clinical side, we also want to think about how can we use the data to populate clinical trials. So really, going from the extreme of clinical care all the way to research. So I'll just briefly mention this first work package and some of the features. So as I mentioned, in New York, we had set up a program, and the program was focused on patients who have advanced cancer of all types. I study prostate cancer, but for this study, we had all cancers. And right now, I think we have over 2,000 patients that we're seeing in this program. These were patients who failed all sorts of various clinical trials and standard of care therapies. And now they were coming for a diagnosis that would use whole exome sequencing, and we developed an analytic pipeline. So this was the starting point of our Swiss program, which was the idea that we would use pipelines that were previously developed and also reporting structures that we already had to think about how we would use that. But we recognized that it would have to be changed for Switzerland. And I think it's going to be, in fact, I think it's going to be entirely new, basically. But that was the starting point. So one of the descriptions that we had in the initial grant application, which I think is still very true, is that at all the different hospitals, and we just depicted a few different types of sequencers. So it doesn't really, matter what the test is, as long as it's getting the right information for the individual patient. But for the perspective of trying to gather all the data together, it does matter, because we have to figure out what the assay is. Did they just look at the tumor cells? Do they look at the tumor and the normal? And if they have the normal, that means you also have germline information. And how do they report it? And so very often, reports are very different. And we even have sometimes in reports, a clinician is given a very complex report and then simply simplifies it by saying no mutations were found or EGFR mutation identified. And that really erases all of the sophisticated data that we've gathered. So it comes all boiled down to a handwritten document that's put in the records as a PDF. So the question is, how could we actually take all of this data directly and put it into a pipeline that would allow us to come up with uniform reporting? And the importance of the uniform reporting is not that we're saying that one site doesn't know how to analyze the data, the other site knows how to analyze the data. The point was that we wanted to have a pipeline where we could say that when we put all the cases through the pipeline, at least we'd be using the same rules. And from a clinical perspective, that's important, because if you have a report, and this might be a level of detail you might not want to know about, but if you have a report, there may be a cut point where you no longer are going to call a mutation. So imagine you have a sequencer that looks at a tumor and you sequence all of the reads, and let's say you have 100 reads for that tumor, and only 3 or 4 of them have something that looks like a mutation. In clinical settings, you may not use that information because it's too low. And so in New York State, where I was working before, it had to be 10% of the reads, so 10 of the 100 would have to have that mutation. If it was lower than that, you wouldn't call it. So in one institution, you might mention only 3 reads demonstrate the mutation, and then you at least know that information. But in most institutions, you would just say it's negative. And so that doesn't entirely tell you what's going on, because imagine that you have a tumor, but when you did the biopsy, you only took a tiny, tiny portion of that tumor, and the rest of it was benign tissue surrounding that. So now, if it's a BRAF mutation, if you find any evidence of that mutation, you know it's a tumor. So you might want to change your rules. And so you can imagine changing your rules. And this brings up probably a great opportunity for thinking about how to change the rules in a way that are still clinically appropriate. So if you've seen something before and you know that at 3% it would also count, you can't just have a rule that says only things at 10% count. So that's something to think about. It's a little bit more detailed than you might want, but that's some of the detail we have to think about. When we looked at the different sites, we asked, okay, what were the assays and how uniform is genomic testing in Switzerland? And if I looked at 3 hospitals in the United States, it would be the exact same thing, which is most of the institutions have many tests with different assays, and the companies are constantly changing their assays. So we need to know not just, oh, there was genomic testing done, but what was the assay? What was the composition of the assay? And so this is important for our database. We have to know exactly what that was, what version. So, maybe in year 1, they didn't have a specific variant, so they did the testing, but they could not have detected an important mutation because they didn't test for it. So you have to know what are the pertinent positives and what are the pertinent negatives. And that's something that is really critical. So in our program, what we're trying to do then is to take the data, whether it's from targeted or whole exome. Most of the data in Switzerland is targeted sequencing, and we can discuss what that actually means if it's not clear. And then take what I would say, quote unquote, is the raw data, which are files that are generated from the systems. So it's not entirely raw data, but then we put the data then into a pipeline, and I'll describe that in a moment. And that will filter out what are called variants, or what we might call mutations that are abnormal. So you may, in a population, you may find them, and they're abnormal if you find them in one form. There are things called SNPs, which are alterations that we see just that describe the diversity of populations. But these are actually alterations that describe something malignant or is known to be deleterious. So this is the overview of the pipeline, and this was developed by one of the computational people that we hired as part of the program. So Federico Comilio has been developing this in Bern. And the idea is to take the raw files, as I mentioned, to do this pre-processing, and then it leaves you with some variants, and these are the mutations. And then we go into a post-processing phase and then reporting. And so I'll just show you what the pre-processing and post-processing actually means. I'm not an expert in computational biology, so I'm presenting this to you. I mean, I can't answer all the details about how this is actually done. But the sequencing data that we receive is, as I mentioned, not uniform. So we may have a test that only looks at tumor. We might have a test that has a paired tumor normal. And I would argue that we probably should be moving towards always looking at tumor normal. And there's a big discussion we could have about testing for germline. That's very different than what we're doing. We're really asking to pair the tumor and tumor-related genes and what is happening in the normal. And the reason why we need that is because patients may have a variant that's just a normal variant, and it may be called as mutation. And patients may also have germline alterations such as BRCA mutations. And that's extremely important to understand, because it has implications not just for that patient, but for the entire family of that patient. And obviously that gets into a whole discussion about the ethics of it, but then also genetic counseling. And that's a really important topic related to precision oncology. So one of the things that you have to do when you create a pipeline, and you might not want to know about this, is that you have to figure out, well, how am I going to actually make a mutation call? And you would think it's fairly straightforward. There's probably an algorithm out there that you could use, and it would align your little piece of DNA to the standard reference genome, and that would tell you what the mutation is. Well, it turns out that that's not so easy, and we can go and describe in the best institutions in the world that do this, there's no one algorithm that perfectly does it. And the reason being is that when you come up with an algorithm that's really good at calling one thing, like point mutations, it may be really bad at calling something else, like deletions or insertions. So these are different types of alterations that occur in the DNA. So the problem is, how do you deal with that? So one way is to come up with a bunch of different algorithms and use them all, and then ask the question, if all the algorithms or most of the algorithms call this as a mutation or an alteration, then we should accept it. And you could make an argument that it's good or bad, but that's the approach we took. It's a fairly conservative approach. One of the problems is that if you're in the research world, so before I was in New York, I was at the Broad in Boston, and it's a large genome center, and the threshold for calling mutations is much, much lower. So in a typical case, you might have thousands of mutations that are called, but from a clinical setting or perspective, that doesn't make sense. So that's why using very conservative approaches for clinical grade calling is important, because you only really want to focus on a small list of the most probable mutations that are driver mutations. So this pipeline then can give us answers with regard to somatic alterations. And we've indicated here that we have what's called a LIMS system, so a lab informatics system that you could use. And this can allow you to pair metadata. And so, as I mentioned, many different tests are used. So you might want to have as metadata what the test was, what the version was, when it was done, what the assay was, and so on. These are important things if you're running a lab because you have to keep all that information. So then the post-processing part, so once you have your mutations, it's going to then also be important to think about, okay, so what are we going to do with that information? And I'll just give you 2 or 3 examples. So one is that if you have a collection of mutations, you may be looking, let's say, for a mutation that tells you that there's a microsatellite instability. So in the old days, we might look at 5 markers. More recently, there's a broader panel. But if you have a lot of genomic data, you could say, let's look across the entire genome and see if there's a genomic signature. So more like a fingerprint as opposed to just like one very focused assay. So thinking about molecular signatures is a possibility. So we can include that in how we analyze the data. We can also think about how we want to present the data. And so I'll show you an example of something called SeeBio Portal, where we can take all the data and export it into a program that allows you to visualize the data. And at the end of this, we hope to generate a report. In Switzerland, it has to be in German and in French and Italian. I don't think we have to do Retro-Romanish. That's not necessary, and probably English. And there's a very nice example I'll just show you from the pathology groups called Pathology, where they've established this as a sort of a standard. And I think it's a very nice approach where you can basically just hover over the language and it'll switch the language. Now, that requires that somebody up front has thought about what is the correct translation. This seems like a very simple thing. You would expect that in medicine or in the hospital, we use the exact same language, but it's actually wrong. And so we, in New York, when we started doing this, we said, okay, we're going to create a simple annotation. And let's say it's a prostate cancer. There's only going to be one terminology for the prostate cancer. Turns out you could have 10, 15 different ways of saying it, depending on who wrote the report. And so what happens is that if you want to do this in some automated way, it's very difficult. So that's why we're moving towards what are called synoptic reports in pathology, meaning that you can only choose, it's like a menu, you can only choose certain things from the menu. You can write a note at the end, you can say whatever you like, but you can only use a certain synoptic form. And that makes the translation a lot easier. So data transfer is obviously very important. And this gets into, I think, critical questions about how do you take patient data or patient-related data and transfer it outside of a hospital? How do you share it? And that's one of our, I think it's one of the main missions of the SPHN program to do that. And I think whenever we engage in conversations at the very start, the understanding should be that we do this in a way that's protecting patient care, patient privacy. So any discussion we have about it should make those assumptions up front, that anything we're doing should take care of that. One way we've dealt with that in research has been that whenever we get the genomic data, we only have a research identifier associated with it. So nobody in the laboratory has any information related to that patient. People make the argument that just the fact that you have the genomic data alone allows you to figure out who that patient is. But I would say that that is something that as part of the data access. So if you have access to the data, there have to be some understandings or agreements. And one of the agreements is that you don't do something like that. So that would be like trying to break into a bank or rob a store. That's not what you're supposed to do with the data. And so I think it is always possible. There's no 100% safe system, but the idea would be that you have good security measures that include probably legal measures to make sure that people would be penalized obviously heavily because obviously it can have an effect on patients. So in this setting that we have here, we're transferring data. This is obviously a subway map that's too complex to read at this point, but the idea is that we'll have a local network, and that works fine for our institution. But then how do we share this across Switzerland? And so Biomed IT is highlighted here as one example of a network that allows us to not only place data, but then also the programs that we're using. And so that's just something that we're working on establishing. And I think it's new to everybody. So how we do this is something that we're all learning. I think it's an exciting process. And just to take a step back again, when I think about the US program that I was involved in, we were trying to do this across the United States. And I think it was virtually impossible to imagine that just because the diversity of systems. I think here in Switzerland, we have a much better chance because we're smaller. And I think this could be a great model system for how other countries could think about doing it. Once you get to a certain size, it just becomes extremely difficult to do something like this. Just a short mention that anything that we're doing, we have to have ethical approval. And we were amazed at how complex this process was in Switzerland. So we're hoping that one of the lessons that we learn and that other members of the SPHN program will learn is how do we come up with good ethical ways that we don't have to keep writing these complex ethical protocols. The ethical protocols are going to be very similar, but with slight changes related to projects. And we went through, I think, 4 revisions in order to get this approved. Now, it is very concerning if your job is to evaluate whether this is ethically correct, or are you concerned, are you ready to turn this program loose. You're sharing data from multiple university hospitals, and you're going to be doing research on it. So obviously, we had to make sure that all of the measures were in place. Are we using the correct approaches for gathering the data, and do we have consents, et cetera? So it's a very complex component. It's not technical per se, but it's something that we really need to think about. And where are we going? So we're really at the beginning, and one of the first things we decided was very low hurdle was to try to gather all of the genomic data from one cancer type that we could, as a first round, run it through our analysis program. And here's just real world data, the types of numbers we're gathering. Now, I would argue that if we got 10 cases from each institution, that would be the same as if we got 1,000, because the next step, the scaling is trivial. But the real hard part is just getting everybody to agree to give 10 cases. And then the next day, we'll have 100 and 1,000. So I don't think the numbers are of concern, that it's a low number. And what we're really hoping then is that we run it through the program, and then we'll be able to compare what was actually called at the institution and what the pipeline calls. Now, there will be discrepancies, so we know that, but it'll allow us to learn in an iterative manner how we can improve the analysis pipeline. First for research, but then eventually I would imagine this might become interesting for us to use clinically. Just briefly about the other work packages. So, it's really nice to have a report that has all the information. So here at the Englander in New York, we had pathology information, we had a summary of the clinical and genomic information, and then we made the decision that we would include all of the data. And so that's a difficult decision to make, because at some point you have to draw a line, and the data in the report is no longer for clinical use. Probably in Switzerland we won't do that, but the reason we did that was because we wanted to make sure we maintain that data, and then for tumor boards, when we talk about the patients, we would have all that information available. The report that we generated, which I hope will be similar to what we'll do here, is a report where you could actually click on the information. So it's a PDF, but you could also use this and click on it, and it'll provide you the actual data that's behind the call. So if, let's say, we say there's a mutation, it would actually show you in a program called IGV, which is a web-based program, where you could actually see what is the data that supports that call. So that's, to me, very important. I mentioned pathology. So pathology has been working on a similar approach. How do you unify all of the pathology data? And this is a program led by Holger Moch and others at ETH, at the hospital in Zurich, University Hospital. And what they're doing is, as I mentioned, gathering a language that can be used across Switzerland that says, for a specific cancer, we use only a certain terminology, so it allows us to share that information. And we feel that both the genomics team and also the oncology team, we're going to share this experience, and then working together, we'll have a very nice system where we have the pathology, genomics, and also pathology together. And I just mentioned we're going to be demonstrating how you can visualize data, and this is work led by Gunnar Retsch's group at the ETH in Zurich. And this is what's called SeeBio Portal, and we also have the SVIP team that's working also to visually present the variants. So we don't have to go into details, but I just wanted to highlight that for researchers, it's really important to be able to have like a Google where you can say, okay, I'm interested in this gene, and it might be really obscure and nobody else in the world is interested in it, but that's what you do. And by using the system, you can just Google that gene, and if it exists, you can find out, okay, what's known about it. And as we populate our database, we can also think about how that's useful in Switzerland, but then also we populate this with publicly available data as well, so you can compare other populations, other studies, and this is, I think, going to be an important tool. It already works, so we're not creating anything new, and I think that's an advantage of where we are now as opposed to where we might have been 5 years ago when it wasn't really robust and up and running, and I think that's really important. So this is just where we are. I think I've mentioned it. I think we're starting to do the first proof of principle studies. We've got the ethics approval. Just a few words about the PHRT program. So we wanted to ask a specific question, and in medical oncology, one of the exciting areas, and Nobel Prize from the previous year was given for immuno-oncology, so understanding how to treat cancer with immune blocking agents. And one of the ideas was that we would try to pair the genomic data with clinical trials in immunotherapy to understand why some patients respond and others don't. So I won't go into all the details. It was just to say that this program is ongoing. And one of the goals that we have is that as they move forward asking scientific questions, we'll also be able to process the data. And in this instance, we'll be using whole genome data, whole exome data, as well as targeted data. And then we'll be able to then compare that, but also hopefully put that type of data into the larger database for other researchers to use that data. So I think I'll stop with that and so this is just a summary. Maybe I'll just leave it at that and just say that we have a lot of institutions that were involved. So this has been a great opportunity for an American to travel around Switzerland. So that's been good. And these are all the individuals. I won't name everybody. I've named some of the key people, but it's been a great opportunity to work with people. And I'm happy to discuss what I've just described for the next few minutes or however long you want to do that. So thank you.